बिस्मिल्लाहिर्रहमानिर्रहीम। अस्सलाम वालेकुम स्टूडेंट्स ऑफ क्लास 9th। कैसे हैं आप लोग? होप ऑल ऑफ यू विल बी फाइन और फ्रेश होंगे आफ्टर अ ब्रेक। सो लेट अस स्टार्ट our lecture, which is lecture number 15, and it is about errors in mitosis. First we will repeat the mitosis because few of the students mentioned that in the lecture number 13 the last phase of the karyokinesis, that is telophase, was missing. So it was by chance. So we will study this telophase and we revise the first three phases also. First of all, mitosis is the division in which the cells divides into two cells from a single parent cell, and each cell, which is called as daughter cell, has the same number of chromosomes which the parent cell has. The mitosis is divided into two major phases. The first is the division of nucleus and the second is the division of cytoplasm. Division of nucleus is called karyokinesis and division of cytoplasm is called as cytokinesis. So karyokinesis is further divided into prophase, metaphase, anaphase and telophase. In the prophase, we studied that the nucleoli which was present in the cell disappears in the prophase. The nuclear envelope which is present in the parent cell, and this nuclear envelope disintegrates or disappears at the end of the prophase. The spindle fibers originates from the centrosomes, and the centrosomes divide into two. They starts to migrate towards the opposite poles, and they originate the spindle fibers also from the protein which is present in the cytoplasm. The chromatin material starts to condense and coil to form a chromosomes. So in the next phase, the kinetochore of the chromosomes attaches with the kinetochore of the spindle fibers, which is known as metaphase, and the chromosomes arrange themselves at the center, which is called as equatorial plate. Now in the next phase, which is called anaphase, the chromosomes which were attached with the each chromosomes which was attached with the two spindle fibers, they starts feeling a pulling force towards the opposite pole, which stretches them and separate them into two. So the chromosomes, each chromosome which was originally present with the two sister chromatids, now this chromosome is separated and the sister chromatids started to migrate towards the opposite pole. In the telophase, as a result of the anaphase, the chromosome is now succeeded in separating identical copies of chromosomes into two groups. But still the cell is attached because the cytoplasm is yet to be divided. Now if we see the telophase and we compare it with the prophase, the telophase is exactly the reversal of prophase. In the prophase, the nucleoli disappears. In the telophase, the nucleoli reappears. In the prophase, the nuclear envelope disappears. In the telophase, the nuclear membrane reappears. In the prophase, the chromatin material condenses from thread-like form to the chromosomes shape. Over here, the chromatin material, the chromosomes uncoil, and after the uncoiling, it is again in the loose thread-like form, which is called as chromatin. So prophase is exactly the reversal of the telophase, or you can say that telophase is exactly the reversal of prophase. Then after that, the division of cytoplasm takes place, which is known as cytokinesis. So in this way, the mitosis completes. Then we studied about the significance of mitosis. That is, this mitosis is very much of importance in the development, and this is a development and then the further growth, that is the further upbringing, growth of the baby. The cell replacement, like the skin cells, the hair cells, and the mucosal cells, the blood cells, the regeneration of the lost parts, like in the sea stars and in the lizards that we see, that replaces its tail in our homes. Asexual reproductions, in which the single cells develop a mass called bud, and the mitosis in the bud develops completely a new plant, or the vegetative propagation also takes place due to the mitosis. So all these are the significance of mitosis. Now we studied that mitosis is a highly regulated process, but when the control of or the regulation of mitosis get disturbed, the number of mitosis and the timing of the mitosis also get disturbed. So after this loss of control, the cells divides uncontrollable, and before in original or in the normal stages, when it divides in a time 4 times, in the abnormal condition it divides multiple of 4 times. So as a result, abnormal growth or tumor develops, which like a mass of cells. So as a result of mutations in the genes, the control center get disturbed and the cells divides uncontrollable to form a tumors. Now there are two types of tumors: benign tumors and malignant tumors. When the tumor stays at the original location and it does not get spread in the other parts of the body, it is called as benign tumor. In the other case, the tumor does not stay at the original location, or rather it spreads to the other parts of the body and causes the tumors or the cancer or the spread of cancer to the other part of the body. This tumor is called as malignant tumors. And the spread of this malignancy or the cancer cells to the other parts of the body is called as metastasis. Hope so, students, it will be clear to your minds. And the skip portion, which is the telophase, is also studied in this lecture. Now memorize it and write it down in your neat notebooks. Thank you. Allah Hafiz.